the Science Guy. Bill Nye, the Science Guy. Bill.
Hi, Michelle.
Hi.
Did you know that the Cheez-Its you're eating have already begun to break down before they even reach your stomach?
Really? How?
Well, there's enzymes in your saliva that break down starch in the Cheez-Its.
Cool. Tell me more.
Well, there's a lot more to it. Let's take a look. Hi there. Bill Nye couldn't make it again, but I'm Sharon's the Science Gal. And today we're going to talk about the chemical breakdown of food. Here to help me are Kristen and Christina. And they're going to be here to help me visualize some important concepts. Digestion is the catabolic process that breaks down large food molecules to monomers small enough to be absorbed by the GI tract lining. The enzymatic chemical breakdown of any food molecules is hydrolysis because it involves adding a water molecule to each molecular bond to be broken. Four major molecules that are broken down are carbohydrates, proteins, lipids, and nucleic acids. The monomers of carbohydrates are monosaccharides. Three monosaccharides that are common in our diet are glucose, fructose, and galactose. Saccharidase enzymes are required to break down complex sugars into simpler ones, such as glucose, which is utilized for ATP synthesis. This can be shown by salivary amylase, which is an enzyme that breaks down starch into simple sugars using hydrolysis. If starchy foods escape being broken down by salivary amylase, they are acted upon by pancreatic amylase in the small intestines. Glucose and galactose are shuttled by secondary active transport into epithelial cells. Proteins are digested into amino acids. This process begins in the stomach when pepsinogen, secreted by the chief cells, is activated to pepsin in the presence of hydrochloric acid. Other enzymes involved in protein degradation are trypsin and chymotrypsin, which are secreted by the pancreas. Although lingual lipase is present in the oral cavity, the small intestine is the primary site of lipid digestion. Because lipids are insoluble in water, these molecules require bile salts to be digested and absorbed. Bile salts are used to emulsify fats by surrounding each fat droplet. This does not break chemical bonds, it just reduces the attraction between fat molecules, allowing easier access for pancreatic lipases. Pancreatic lipases catalyze the breakdown of fats by cleaving off two of the fatty acid chains, yielding monoglycerides. The nuclei of cells of ingested food contain DNA and RNA. These structures are degraded by pancreatic nucleases. Intestinal brush border enzymes then break the nucleotides apart. Whether it is carbohydrates, proteins, lipids, or nucleic acids being broken down, the monomers are absorbed into the bloodstream and then transported via the hepatic portal vein to the liver. The ability of our digestive system to break down and absorb nutrients is vital to our survival.
Suffer first to the storm.